Now let's look at the somewhat difficult task of identifying the various cells in the erythrocyte lineage and in the neutrophil lineage. We'll start with the erythrocyte lineage. As you do this, I suggest that you keep figure 6 in your notes open next to you, or if you have a textbook, you might want to open it onto a page which shows both of these lineages. The first recognizable cell in the erythrocyte lineage is the proerythroblast. And one way to recognize the erythroblast series is that they all have this speckled type of nucleus up until you get down to the cell labeled 4 here, the normoblast. And they generally have a basophilic cytoplasm surrounding this large nucleus. The proerythroblast has a large round nucleus with lots of nucleoli, which is what gives it this speckled appearance, and a bluish cytoplasm due to the presence of large amounts of RNA. As this proerythroblast morphs into the next stage down in the lineage, the basophilic erythroblast, the nucleus stays pretty much the same, but the cytoplasm becomes bluer and bluer due to the synthesis of more RNA in preparation for synthesizing large amounts of hemoglobin. As this basophilic erythroblast goes through cell divisions, its daughter cells, the polychromatophilic erythroblasts, get to be smaller and smaller, but they still have this checkerboard or speckled nucleus shown here and shown here. And remember that this is a transition. There are no sharp dividing lines. So these polychromatophilic erythroblasts, as they divide, and they're still able to divide, they become smaller and smaller, and they start to put out large amounts of hemoglobin, so that the cytoplasm here becomes more eosinophilic. And when this eosinophilic hemoglobin mixes with the basophilic RNA, one gets this transition from a basophilic cytoplasm to a more eosinophilic cytoplasm, and so this is called a many chromatophilic erythroblast. The polychromatophilic erythroblast eventually stops synthesizing RNA, stops synthesizing hemoglobin. Its nucleus becomes pyknotic, becomes very dense, loses this speckled appearance, and the next stage down from this normoblast or orthochromatophilic erythroblast will be when this cell ejects the nucleus, becoming a reticulocyte, which is not shown here. Reticulocytes still contain a little bit of RNA, and once that RNA is lost, it becomes a bag full of hemoglobin, takes on the typical shape of an erythrocyte, and loses all of its basophilia. On the right hand side here I've shown some stages of the transition of a polychromatophilic erythroblast. As it becomes smaller and smaller, its cytoplasm becomes less basophilic, more pinkish due to the buildup of hemoglobin. The nucleus becomes smaller and smaller, and eventually the nucleus becomes pyknotic. The cytoplasm becomes pretty pinkish due to the large amounts of hemoglobin. The nucleus will be ejected, resulting in a reticulocyte, and once the little bit of RNA that is still left in the reticulocyte is destroyed, the cell will become a full-fledged erythrocyte. In looking for these stages of erythrocyte development, I find it easiest to look around until I find the basophilic erythroblast. This is fairly easy to see with this large speckled nucleus, perhaps shown a little better here, very intense cytoplasm, basophilic cytoplasm, and then look around until you find something that looks fairly similar but has a lighter, a less basophilic cytoplasm, and that will be the first visible stage of this lineage, the proerythroblast. And then look around until you find something that has a smaller nucleus, a cytoplasm that is not quite as basophilic as it is here due to the production of hemoglobin. That would be the polychromatophilic erythroblast. And then the normoblast or orthochromatophilic erythroblast is fairly easy to see because it's got this very dark nucleus, shown possibly better here. And as I said, eventually that nucleus will be ejected to form a reticulocyte, which we don't see, and then of course a full-fledged erythrocyte. The first recognizable stage in the neutrophil series is the myeloblast, and in the myeloblast stage the nucleus is rounded or slightly bean-shaped. The cytoplasm has this dirty, washed out gray appearance, but what is characteristic is that there are no granules in it, and unlike the erythroblasts, the cytoplasm does not have a lot of blue color due to the presence of ribosomes. Now, because we're looking at the neutrophil stage, the granules are not going to be as prominent as they would be if we were looking at the eosinophil stage. The next step in the development of the neutrophil lineage is the transition into the promyelocyte stage. The nucleus is still somewhat round to bean-shaped, but one can start to see the presence of these nonspecific azurophilic granules in the cytoplasm. As we transit from the promyelocyte stage to the myelocyte stage, shown here, the cell becomes smaller due to division, and the nucleus becomes a bit more bean-shaped, and there is still visible granules present in the cytoplasm. The myelocyte stage is the last stage that can still divide, and the myelocyte stage transits into the metamyelocyte stage. The nucleus has become more bean-shaped. In this particular cell that I'm looking at, there are relatively few visible granules, and the metamyelocyte stage is no longer capable of division. The metamyelocyte stage then passes through a stage called the stab cell or band cell. Here it is, number 5 here, where the nucleus becomes distinctly horseshoe-shaped and actually is starting to lobulate, as is seen here. With further development from a stab cell to a mature neutrophil, one sometimes sees these early mature neutrophils, but really the stage that I would want you to look at would be the transition to a mature neutrophil with a multilobulated nucleus joined together by these strands of chromatin. Megakaryocytes are very easy to recognize. They're giant cells. They're derived from megakaryoblasts under the influence of thrombopoietin, and they undergo endomitosis, a process by which DNA replication occurs, but the nucleus doesn't divide, the cell doesn't divide. So they result in these huge polyploid cells with a DNA content, as I say in the notes, of 64N or even greater. What you need to know is that these megakaryocytes extend long branching processes called proplatelets between the endothelial cells which line the sinusoids of bone marrow, and platelets are pinched off from the tips of these processes. It is important for you to realize that a single pluripotent, or multipotent is another word for it, stem cell gives rise to all of the cell types that you can see in bone marrow. How does this happen? Well, as I said in the notes, it is due to the effect of paracrine and endocrine growth factors, the colony stimulating factors, which are produced by the stromal cells, by macrophages, connective tissue of bone marrow, and by other organs outside of the bone marrow, such as erythropoietin is produced by the kidney and it stimulates the erythrocyte lineage. Thrombopoietin is produced by the kidney and the liver, and that stimulates the division of the megakaryocyte cell lineage. Granulocyte macrophage colony stimulating factor stimulates the progenitor cells for granulocytes and the monocyte cell line. And then later on, other colony stimulating factors are required for the further development of all the lineages.